Hello, and welcome back to the Sound of Science AP edition. Today we're going to be talking about tonicity and osmosis. And if you're sitting there thinking, I have no idea what tonicity is, don't worry, you're going to know in a couple of minutes exactly what it is. But the key point here is that this is all dealing with the movement of water into or out of this. Osmosis is literally the diffusion of water across a semi-permeable membrane or a selectively permeable membrane. When large amounts of water move back and forth across the membrane, because water is polar, because it's larger molecule, it needs to move through a channel called an aquaporin. And that's something that we talked about in a previous video. Osmolarity, which is the total solute concentration in a solution, is actually really, really important to how much water moves and how quickly it moves through these aquaporins. So, just a couple of other definitions you need to know. A solvent is what's doing the dissolving. Water, in this case, would be the solvent, usually the liquid. Solute is what's being dissolved. This is something like a salt. The solution is what you get when you put the two of them together. So the solvent and solute mixed together make salt water. That's your solution. Now let's get to that tonicity. This is essentially the measure of the relative concentrations of solute between two solutions. So essentially, you are looking at concentration both inside and outside of the cell in this situation, mainly because we're just dealing with biology, we're dealing with living systems. So, this is going to have an effect on the cell's physiology, on its physical characteristics. It also allows for growth and development of cells, and that's just the movement of water, the movement of molecules in and out of the cell is going to affect whether or not or how the cell is able to develop and how it's able to grow. So here's a couple more words for you to learn. Hypertonic, isotonic, and hypotonic, which you may remember from high school biology. These words should not be completely unfamiliar to you. If they are, don't panic. You're going to learn them now. So hypertonic solutions or hypertonic environments have more solute, less solvent. So in this case, the environment is going to have more solute in it, less solvent. So it's a higher concentration, which means that water is actually going to move out of the cell. And we'll explain all that in a minute. Isotonic means that they're fairly equal. It's an equal concentration inside and outside. And so there's no net movement of water. It's going to move back and forth pretty freely, just like you see in the diagram. Hypotonic has less solute and more solvent. So think of this as like it's waterlogged. It's got lots and lots of solvent. And so in that case, the water is actually going to move into the cell. And all of this is an effort to reach that isotonic state. I'm going to read this to make sure I say it exactly correctly. Water moves from an area of high water concentration or low solute concentration to an area of low water concentration, which has high solute concentration. Okay. I want you guys to think of it this way. If we have high water concentration, that automatically means we have a lower solute concentration. If we have a low water concentration, we automatically have a higher solute concentration because the two are connected, right? Remember, concentration is the amount of solute per volume of solvent. And so if we increase one, we decrease the other, and vice versa. And I want you to kind of think of it doing this, okay? So solutes are going to continue to diffuse along their concentration gradients as they are able. But remember, there are certain items, certain materials that are unable to move into or out of the cell through the membrane without help. So some things are not going to move at all. In an isotonic environment, you're going to have dynamic equilibrium. And this is just a case where water moves in and out. Essentially pretty freely. It's going to go back and forth because they're equal. There's no gradient there. So if there's no gradient, if they're pretty equal, water just kind of flows back and forth. So take a look at this diagram. And my question for you is this a situation where we're talking about something being hypertonic or hypotonic? So if you look at this diagram, first of all, you need to figure out where the high water concentration is. And in this case, the high water concentration seems to be outside of the cells, which means that the low water concentration is going to be inside of the cells, which means that water is going to move in the direction it needs to in order to reach that equilibrium. So, water is actually going to move from the outside to the inside of the cell. In that case, we are talking about hypotonic. So, osmoregulation, how does this work? This is the same basic idea, but we're going to talk about it in plant cells. So, plant cells have to maintain internal concentrations at specific levels. Otherwise, certain processes won't work. And they also need to maintain water balances. So, in a situation where you have hypertonicity, water is going to be moving out. We refer to this as plasmolysis. And yes, you do need to know all these terms. Hypertonicity, I always think of hyper like hyper kids, right? Everybody talks about, oh, that kid's so hyper. That kid's, they just want to run around. They want to be like outside running all the time. So, hypertonicity, I always get this mental image of kids like running out the door, just like water is moving out of the cell where it has more space to be, where there's more things happening. In fact, there's more solute happening. So, plasmolysis is what happens when you're in a hypertonic environment. Isotonicity, no net movement. And I always just remember that ISO, to me, I always think ISO means kind of equal or the same. We refer to that as flaccid, and that's just where the water is moving back and forth pretty freely through the cell membrane or the aquaporins. Hypotonicity, that's where water is moving in. And I always think of hypo and I think of the O in it. Hypotonicity means water's going to flood in and the cell is going to grow and look like an O if we're talking animal cells. Plant cells, obviously, that's not the case, but it's just my trick for how I remember it. So, hypotonicity, the cell's going to grow. Water, because water's moving in like a water balloon. In this situation, we would actually refer to that as being turgid. Turgid is a fantastic word. Make sure you know it. Cell walls actually also help with this osmoregulation. Essentially, plant cells kind of live in a state of hypotonicity. That is where they are happiest. Water moves in, it increases that internal water pressure until the vacuole inside that plant cell has grown, hypo, to the point that it's pushing back against the cell wall. This makes the plant turgid and it helps maintain the plant structure as well as all of the other things that water is required for, these concentrations are required for. I want you to think of this like if I had a cardboard box and I had a basketball that just fits in the cardboard box when it's full of air. So I put that basketball in there and I put the cardboard box in there and I try to squish the cardboard box. Well, it won't squish but so far because I've got that basketball in there providing support for that cardboard box. Now, let's say that I deflate that basketball. Now that a basketball is all deflated and shriveled up, and I put it back in the box, now I can move those sides. I can actually squish the box in. This is how the cell wall and the vacuole work together. Think of the vacuole like the basketball, and the cell wall is like the sides of the cardboard box. Animal cells, same idea. We talk about them also going through each of those things. But with the animal cell, because we don't have that cell wall for structure and for support, we actually end up in a situation where cells that are flaccid, that's your kind of your normal state for your animal cells. These are red blood cells. We always like to use these when we talk about tonicity. Not really sure why, except that they show pretty easily what we're trying to see here. If it's in a hypotonic situation and water's moving in, the cell itself can actually lyse. It can burst from too much water. If it's a hypertonic, remember that water is moving out like those hyper kids, and the cell can actually shrivel up. Okay, so how the hell do single celled organisms deal with this water problem? So you've got to imagine if you're a little paramecium, you're an amoeba, and you're floating around in a pretty aqueous environment, and you're taking in water constantly. How do you prevent yourself from lysing? You don't have a cell wall to protect you to keep that from happening. So this is how paramecium figured out to deal with this. If you look at this section right here, I know it's hard to see, this paramecium is using its contractile vacuole. It's literally filling up with water. Water is flooding in. It's in a hypotonic situation. And then it uses its cytoplasm and it contracts that vacuole. It actually squirts water out. If you ever look at these guys live under something like a dissecting scope, um, on a microscope slide, you can actually see this happening and you can see them squirting. It's actually really, really cool. Unfortunately for this paramecium, if we decided to place it in salt water, it would start losing water too quickly and it would actually shrivel. Okay, we ready for a quick math lesson? Water potential. Water potential just measures the tendency of water to move by osmosis. Okay. Think about potential energy. Like, ignore the whole water, ignore pressure for a minute. Think about potential energy and think about how if you have a hill, right? And you have a ball on top of the hill, then that ball has high potential energy. As it starts moving down the hill, it gets higher kinetic energy. But the lower it gets down the hill, the lower potential energy it has. Because that potential energy is essentially determined based on its location, how likely it is to ultimately move. So, water pressure or water potential essentially is the same thing. Where you have that high water potential, that's like high potential energy. Lots and lots of water here, lots of solvent here. And it's going to move to an area that has a lower water concentration or a lower water potential until it's equaled out. So water potential can be positive, it can be negative, or it can actually even be zero. It can be equal. The more negative the water potential, the more likely water will move into the area. So if I have a negative 10 bar water potential, that's going to move pretty quickly into that area. If you have a positive 10 bar water potential on the other side. The key here is movement, usually across a membrane. So keep that in mind as we talk about this. There are some things here that you kind of need to know, you need to memorize, and some things that you're going to find on your AP formula sheet. So, water potential of pure water in an open container is zero. Okay. Increased solute in the water equals increased solute or potential decreased water potential. So the more solute we have in the water, we increase our solute potential, but we decrease our water potential. Increased water potential is equals increased pressure potential. Decreased pressure potential equals decreased water potential. In an open system, pressure potential is going to equal zero. So water potential is the same thing as the solute potential. That part right there is really important for you to remember. In an open system, pressure potential equals zero. So water potential equals the same thing as solute potential. This that I have right down here for you is solute potential equals negative ICRT. I stands for the ionization constant, which for sucrose is 1 and for salt is 2. C is our molar concentration, essentially molarity. Okay, so moles of solute per volume of solution. R is our pressure bars per mole K. T, temperature in Kelvin. And don't forget, temperature in Celsius plus 273 equals Kelvin. So if a tree is planted in soil with a water potential of -4 bars and the roots of the tree has a water potential of -8 bars, in which direction will the water move? Well, it's going to move from an area of higher water potential to an area of lower water potential, rolling down the hill, right? So -4 is actually more than -8. So it's going to move from the soil into the roots of the tree, which is fortunate for the tree. I know how easy that felt. Let's talk about this one. What is the water potential of a zero point 0.5 molar sucrose solution at 21 degrees Celsius in an open system? Okay, threw a lot of numbers at you. For those of you like me that are not super math people, it's okay because this is a formula, and this formula will actually be given to you. Okay, so first we need to solve for water, for solute potential, so which equals negative I CRT. So let's put all of those things together. So we know it's sucrose. Sucrose's constant, ionization constant, is 1. So we have negative times 1. Then we're going to use the molarity. So that's 0.5. Then we're going to use this constant and that R figure, and that's going to be 0.0831. That's the constant. It's going to be that no matter what you're looking at. That is your constant. Plus temperature in Kelvin, so 273 plus 21. If we multiply all that out, make it negative, we're actually going to end up with negative 12.22 bars. Then we need to remember that this is in an open system. So the pressure potential equals 0 in an open system. So in this case, our water potential is going to be a negative 12.22 bars. So you don't need to memorize those, but just understanding how to essentially plug and chug. Plug numbers that we've been given into that formula. So we're going to practice this in class. Don't worry, this is not the last time you're going to see water potential. Or worry, this is not the last time you're going to see water potential. So hopefully this has helped you at least get introduced to the concept of water potential. And on that note, I'll see you next time.